Okay, so you know how to trace what happens with one trait as it goes from generation to generation using a Punnett square. What happens if you want to follow more than one trait simultaneously? Mendel did this. So after he tested the inheritance of one trait, he wanted to see if traits influenced each other. Okay? According to his idea of independent assortment, he figured that because all of these characters, these elementin, were separated from each other during fertilization, or during gametogenesis, and then they recombined during fertilization, he figured that one trait didn't affect the transmission of another trait because they'd be separate from one another, okay? Because they're packaged into different gametes at random. That's part of the laws of segregation. So what we now know is that since we're talking about these things, these alleles or these genes, yes, chromosomes separate during the formation of gametes, and then they recombine during fertilization. And as long as the traits are on separate chromosomes, they will sort independently. If they're on the same chromosome, they may travel together unless they're very far apart from one another on the same chromosome. It turns out that when the chromosomes are getting ready to divide, and they're all squinched up in the center of the cell during what you remember is called metaphase, they're all sort of squished in the middle. Chromosomes that are copies of each other, the pairs, can switch pieces. They switch ends sometimes. It's called crossing over. And so if you have a gene on one end of chromosome 1, for example, and a gene on the other end of chromosome 1, and during metaphase they're all squinched together in the middle of the cell, and the two chromosome 1s next to each other switch an end, they may have just switched one of those two traits with the other copy of the chromosome. And if the allele is spelt differently, then you have a slightly different combination. Crossing over happens randomly, and we don't know when it's going to happen, why it's going to happen. We just know that it does happen. Okay. So his next experiment then was to look at two traits at the same time. So he did one trait, he did all 7 of those traits that we looked at, and he did one at a time, and now he's going to combine them. Okay, and so he took a look at seed shape, round versus wrinkled. Round is capital R, wrinkled is lowercase r, so round is clearly the dominant one. Seed color, yellow or green, we use Y's for that, so you can tell which the dominant one is there. Which one is dominant? Yellow, that's right. So we start with true breeding. True breeding means, it's another way of saying homozygous. Okay, true breeding round and yellow are both dominant, so true breeding round and yellow is homozygous round and yellow, all capital letters. And wrinkled green, that's homozygous recessive, all lowercase letters. Okay, as you might expect, he got all heterozygous people, or plants actually, these aren't people. But it works the same way in all organisms. So he got all dihybrids. They're called dihybrids because they're hybrids for two traits. Okay? And that was what allowed him to figure out that round was dominant and yellow was dominant because green and wrinkled disappeared. Remember, that was his way of telling which was dominant and which was recessive. If he took true breeding for each trait and he mixed them together, the trait that disappeared in the first generation, well, that was your recessive trait. So he was able to figure out that round was dominant and yellow was dominant by doing this first cross, two traits at the same time. Okay? Now he crossed the two dihybrids together. Okay? So 2 of the heterozygous plants are going to be crossed together. Since the F1 is the first generation, F stands for filial, which means offspring, or actually it literally comes from the Latin for son. But F1 means the first generation. After the parents, since they were all dihybrids, so heterozygous for both traits. When they formed gametes, each gamete got either a capital or lowercase for each of those two traits. Okay, and then because of independent assortment, the combinations of, and we're talking two traits in the same cell, so we have to look at one R and one Y for each gamete. And since they each of the parents has a capital and a lowercase, and each gamete has to get one R and one Y, the combinations of R's and Y's that you can have, there's quite a few of them. Okay? You can have capital R, capital Y. And each parent could give a capital R and capital Y. You could have capital R and lowercase r and capital Y, or lowercase r and lowercase y. Those are 4 different combinations of those 2 traits that could exist in each gamete after they've separated. That means your Punnett square is now going to be 4 by 4 instead of just 2 by 2. When you're following the transmission of one trait, there are only 2 alleles and they separate, and so you've got 2 alleles for each parent, so that's a 2 by 2 square. Well, now we're looking at 2 traits simultaneously, and with dihybrids, specifically with the 2 heterozygous traits, we have 4 different possible combinations of 1 R and 1 Y, and that gives you 4 by 4 Punnett square. It looks like this. Okay, now I've color-coded this a little bit so that you can sort of understand the phenotype and genotype ratios here. Okay, anything, remember, that has a capital letter will express the dominant trait. So you'll see that we have 9 squares here. There's sort of a yellowish color that represent at least 1 capital R and 1 capital Y when we put them together. That means that they will have the dominant phenotype for both of those traits. They'll be round and yellow seeds. We've got 9 of those. We have 3 that have at least a capital R, but no capital Y. There are 3 of them that will be round, but have green seeds. There are another 3 of them that will be wrinkled because they don't contain a capital R, but they do contain at least 1 capital Y. And so they'll be yellow, wrinkled and yellow. And then there's one way down here in the lower right-hand corner that has both recessive phenotypes and is a green, wrinkled, seeded plant. Every time you cross 2 dihybrids, you're looking at 2 traits, and you cross them together, you'll always get a 9:3 to 3:1 phenotype ratio. Always. Now this is a big Punnett square. 4 by 4, it may not seem like it's that big. Well, it's only 4 times bigger than the other one, but it's a lot bigger. There's 16 different possibilities, and that's just 2 traits. Imagine if we wanted to find 3 traits. We wanted to follow 3 traits at the same time. That gets very unwieldy. 3 traits would be an 8 by 8 Punnett square, 64 different possibilities. You drive yourself crazy trying to fill them all in. So fortunately, there's an easier way to do it. You can use something called the product rule. So we know about this phenotype ratio: 9 dominant, both dominant traits, 3, 1 dominant, and the other one recessive, another 3 with the opposite combination, and then 1 out of every 16 will be both recessive. Okay, so it's ratios, right? 9 out of 16 are both dominant, 3 out of 16 dominant for color but not dominant for seed wrinkling. Another 3 out of 16 would be the opposite way, not dominant for yellow, but they're green seeds, but then round seeds. And then 1 out of 16 possibilities. The possibility is 1 out of every 16 plants should be both recessive. So there's some ratios there: 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16. Okay, so if you want to figure out, based on this particular cross, 2 dihybrids, what's the chances of getting homozygous recessive for both traits? It's 1 out of 16. Okay, now keep that number in mind, 1 out of 16, because you're going to see how the product rule will get us that answer without doing this giant Punnett square. Okay, you don't have to do it. You can use the product rule. Okay, now the product rule takes each trait separately. Okay, so we start by looking at, well, remember, we were crossing 2 dihybrids. So we were crossing heterozygous for round and heterozygous for yellow seeds. If we treat them all individually as a single trait, so we've got over here on the left, we've got the round trait, right? And we have 2 hybrids, heterozygotes, and we're crossing those, and that's a classic Punnett square, and you know that there is a 1 out of 4 chance of producing a wrinkled seeded plant. Over on the right here, we're looking at the color, okay? And again, it's according to Mendel's cross, he had a hybrid, dihybrids, right? They were hybrids for both traits, heterozygous for both traits. So this is heterozygous versus heterozygous. It's a little hard to tell with the font here, but we have a capital Y and a lowercase y in each condition here. And again, if we want to look at green seeds, it's 1 out of 4. 1 out of 4 possible chance to have a green seeded plant. So if we want to know, following these 2 traits together, what's the chances of having both a wrinkled and green seeded plant? All you have to do is multiply the 2 frequencies from the 2 traits, 1 out of 4 times 1 out of 4, and you get 1 out of 16. which is exactly what we got when we did the 4x4. So this always works. Figure out your chances for each trait and then multiply them together. So let's take this very same example and just leave this particular slide up on the video, but pause the video in just a second. Here's what I want you to figure out. Figure out what, given these two parents, dihybrids, heterozygous for two traits, what are the chances, what is the probability of producing another dihybrid offspring, another a plant that is heterozygous for both traits. Figure out the frequencies, multiply them together, and that should be your answer. Okay, so we're looking for what are the chances that you could produce capital R, lowercase r, capital Y, lowercase y in your offspring. Stop the video, do the calculation, and then come back and we'll see how you did. All set? All right, so if we use the product rule, we say, okay, for round versus wrinkled, the chances of having a hybrid, heterozygous, is 2 out of 4, or 1/2, 1 out of 2. Okay? Over here for the yellow versus green seeds, the chances of having a hybrid, heterozygous, is 2 out of 4, or 1 out of 2. If I multiply those two together, 1/2 times 1/2, I get 1 out of 4. All right? Out of 16, 1/4 is the same as 4 out of 16. So if we go back to the big Punnett square, let's count the ones that have heterozygous for both traits. We've got 1, 2, 3, and 4. 4 out of 16 have capital R, capital Y, small y. That's 1/4. See, it always works. All right? Watch the video again, take good notes, ask questions if you're not sure.